Everyone, in this video we're going to talk about the different structural level of FGFR2 protein. We'll mainly focus on the secondary and tertiary structures of the protein. At the end, I will discuss how does the mutation disrupt the normal function of FGFR2. Before I start, I would like to tell you that the FGFR2 protein is a receptor, and receptors have the function of binding to other proteins. In the case of FGFR2, its binding partner is the FGF1 protein, which is a fibroblast growth factor 1, and FGF1 is involved in many physiological and pathological activities of the body. As mentioned earlier in the website, every protein has a primary structure, which consists of amino acid sequence which looks like the colored beads here. Here we can see the two amino acid sequences. As you can see, they're colored differently, so they represent two different chains, which gradually combine to form the final structure of FGFR2 receptor, which we'll see later. To form the receptor, both the sequence of the chain fold to form a spiral-shaped structures called alpha helix and arrow-like structures called beta sheets. These two are the main secondary structures of any protein, as they define the specific geometric shape of the protein, which is caused by intramolecular and intermolecular hydrogen bonding between the amino acids. Then these chains further fold to assemble the different secondary structure elements, as you can see here, in particular arrangement. This gives the protein its tertiary structure. What you can see in the right is a 3D model of FGFR2 receptor, which is created by folding of 32 beta sheets in total, as there are 16 from each chain, which are colored in yellow here, and 8 alpha helices, as there are 3 from one and 5 from other chain, which are colored in red here. These numbers do not count the regions shown by the blue circles, as they are FGF1 protein. Finally, the quaternary structure occurs when the units of tertiary structure aggregate to form homo or heteromertamers. This structure also comprises of disulfide bonds. What you can see in the extreme right is the actual quaternary structure of FGFR2 with 4 disulfide bonds, 2 from each chain. I've colored the model by chain and shown surface as well, which shows the different components. This is a more simplified version of FGFR2 receptor, where you can see all 3 extracellular domains of the receptor. This model shows FGFR2 in complex with FGF1. So what you're looking at in the model is FGFR2 protein, which is a receptor, binding to its ligand another protein called fibroblast growth factor 1. This interaction activates a signaling cascade which controls bone cell growth. The model only shows part of the full FGFR2 protein, that is, it shows the active domain, which is the part that sticks out of the cell. This model shows FGFR2 in the dimeric form, that is, the two FGFR2 proteins coming together to form the mature receptor. Each FGFR2 is made by a single strand of mRNA that has been processed by the ribosome. We'll discuss about the mutations in the next video.